Hello and welcome to the Logos Research Associates live stream broadcast. I'm Fred Balhoff, an associate and the treasurer for Logos Research Associates, and it is my privilege to be your host for this presentation. Logos Research Associates is a fellowship of scholars and scientists that are committed to upholding a high view of science and a high view of scripture. We sincerely believe that scholarship, logic, and the scientific method can and should be used to show that the biblical historical claims are both credible and ultimately superior to the secular model of origins. Logos sponsors numerous research initiatives and projects involving many different experts and scientific disciplines, all working towards the goal of increasing the explanatory power of the creation model of origins and of Earth history. We'll share more of these exciting research opportunities in tonight's broadcast. We also recognize that as humans, our scientific opinions are fallible and subject to change, and therefore we stress that our viewers and supporters do not place their faith in our organization or any other human authority, but to place ultimate faith in the finished work of Jesus Christ. The goal of this live stream series is to provide you with the opportunity to hear from top researchers who will convey cutting-edge scientific evidence for the creation account. This evening, we'll focus on a particularly exciting frontier in creation research, specifically the discovery of original biochemical remains in supposedly millions of years old fossils. Before I introduce our speaker to begin our presentation, we will open a short prayer, and if you could please join me. Dear Lord, thank you so much for the opportunity to hear from Dr. Thomas tonight. Thank you for those that put together this broadcast and the work that they have done behind the scenes. We pray for all of us that will be listening to the presentation, that you will open our hearts to hear what you have to say through Dr. Thomas, and that you will help us to see new information, the reality of your scripture, and to help our faith grow stronger. Thank you for the minds that you have given these researchers that are part of Logos, that they are using their gifts and talents to help our faith to grow. Thank you for your creation and how wonderful and beautiful it is. We pray these things in the name of Jesus. Amen. Throughout tonight's presentation, we encourage you to type in comments or questions on your live feed through YouTube or Facebook. We'll take some time at the end of the presentation to answer some questions from the audience. I now have the pleasure of introducing our speaker. Joining me tonight is Dr. Brian Thomas, research scientist at the Institute for Creation Research in Dallas, Texas. Dr. Thomas received his master's in biotechnology from Stephen F. Austin State University and his PhD in biochemistry from the University of Liverpool. He has a teaching career in both Christian education as well as teaching in university settings in the Dallas area. Dr. Thomas joined ICR in 2008 as a science writer before taking on the role of research associate in 2019 and research scientist in 2021. He is the author of several books, including Dinosaurs in the Bible, and has contributed several of ICR's publications. His PhD dissertation, Ancient and Fossil Bone Collagen Remnants, which is particularly relevant in light of our topic this evening, is available in book form from ICR. Without further ado, I'd like to welcome Dr. Thomas to the broadcast.
Well, thank you, Fred. I appreciate that introduction, and it's an honor to be linked with and I guess a member of Logos Research Associates as I'm working at the Institute for Creation Research here in Dallas. Yeah, I just wanted to invite everyone into my living room here and appreciate everyone attending and clicking in. And I think what I'll do is I'll just give the nutshell, tiniest version of what I call my creation conversion, and then we'll get right into the topic. So is this the part where I show my slide?
Yes, go ahead.
Yeah, okay. So I'm gonna let's see. We practice this, so it's absolutely seamless and flawless and smooth, right? Well, tonight we're looking at what I'm calling fresh fossils from 4 continents, and but just to let you know what I find stimulating and relevant about this really stems back to my creation conversion, and it was back when I was in college, and I was what I would call a nominal Christian, or a Christian in name. I considered myself a Christian. Went to church sometimes. but as I began to develop a relationship with a new friend there in college, he challenged me about my Christianity, and then he challenged me about my beliefs in evolution, millions of years, and I was convinced that the fossils were millions of years old. That's all I had ever heard, and so anyone who would say that anything other than the mainstream perspective on these fossils, that dinosaurs lived and died in a dinosaur age, like that's just baked into the furniture of the living room of my mind. And to dislodge that took a great deal. And so fortunately I had a friend who was willing to put forth a great deal of effort to challenge me. But the way he did it was pretty brilliant. I mean, he just asked me, um, did he just he proposed this concept that they assign ages, they secular scientists assign ages to the fossils based on the age assignment of the rock layer that the fossils are found in, and that other scientists, geologists, assign ages to the rock layer based on the age assignments of the fossils that are in them. And then he said, did you know that that's circular reasoning and it's bad science? And this blew me away, and I said, surely that's not the case. And he said, okay, well, what is the case? Like, what are they actually measuring so that you know for sure that these are that these represent vast ages long, long ago? And I looked and looked, and he asked and asked, and I finally said, look, I'm sick and tired of you asking me. And he said, I'll make you a deal. You read this creation book and I'll stop bothering you. And I thought, yeah, I'll read the book and I'll refute it, because creation science is total nonsense. And so the book that he had me read was this one right here. I have it still in my living room: Scientific Creationism. What a horrible title. It just said to me, don't even read me unless you want to refute me. So I opened it up and started reading, and I had a pen and paper in hand, ready to write down all my notes about how dumb the book was. And the more I read, the more I liked it. And actually, I never found a flaw in it, and I just kept reading, and I just thought, no one's ever told me this. And and then I realized that, like, my professors at the university weren't even telling me about the great design, about these fossils that are found in water burials, waterborne death assemblages. And it's some information that I just didn't have. But after this book, which covers everything from the organization in the cosmos, which began to convince me maybe the cosmos, maybe the stars are organized instead of just random. I mean, if it's really random, why isn't the universe just a big ball of energy instead of organized into atoms and stars and galaxies and galactic clusters, etc.? Like, that's a lot of organization. And so by the time I finished that book, I just thought, man, I have been lied to. And then my view of the Bible and of my origins in Genesis really skyrocketed. And so then I began to read Genesis with fresh eyes, and I became convinced, based on this book and other things I began to read, that the history that we have in our Bibles explains the real world better than the mainstream model of human history and of the world's origins. Much better. So that my view of God himself elevated, because now he becomes the God who wrote down accurately how it really happened. And I can trust him personally because I can trust what he wrote down through his prophets. And it's been my passion ever since, really, to explore these issues and to try to encourage Christians, especially, that you're on the right track if you're taking Genesis history straightforwardly. So that's why I got into this whole discipline of fresh looking fossils. And I just think that if Christians knew more about these, maybe from four continents, that we'd be confirmed in the idea that these fossils and these rock layers really do look like Noah's flood deposited them. So I'm content, very content, in the defensibility of the flood model for what we're about to see. This is the first publication that caught my eye. It was 2008. I didn't get around to seeing it until probably 2006. But these published in the journal Science. And so you could see in box A, there's a little bit of connective tissue, and box C, there's a lot more connective tissue, even strands of collagen-based connective tissue fibers, like whole tissues still intact. This is all liberated from a Tyrannosaurus rex thigh bone, a femur. And of course, the mainstream scientists, many of them anyways, uh, cried foul, and they knew that this could not be from the T-Rex, because they understood the biochemical decay rates. So biochemicals decay much too fast for them to still be present in bones or in any setting that's more than something like 1 million years old. And these are 70 million. That's the age assignment, and that's really not negotiable within that worldview. And so we've had this game of ever since, of trying to figure out how can we get these materials to last as long as we need them to, in order to fit the worldview. And so the more I look, the more I wanted to find, is this just one T-Rex or is it more? And how is it distributed? Well, as the title of our talk indicates, we're on at least 4 continents, and growing. So here's a model of collagen. This shows the position of every atom. As you can see on the top, there's a fully grown, I guess, fresh collagen manufactured there, and it's baked within the bone. It's also within, of course, connective tissue, cartilage, like what makes up your nose. Skin has a large degree of collagen in it. Subtle differences to the collagens in those different areas of the body, but all vertebrates make it, and bacteria don't. Um, but over time, this illustration shows over time, even this highly organized collagen made of 3 strands, they're just artificially colored green, blue, and silver. Um, they do fall apart because chemistry happens according to the second law of thermodynamics, which describes how everything that's highly organized becomes disorganized with time, unless something comes along and reorganizes it. So this does become disorganized as soon as a body dies. The collagen and everything else in that body begins this decay process. The reason our bodies don't fall apart during our lifetimes is because we have bone cells constantly replenishing our bone and skin. I think we go through our skin. You have a new suit of skin every, I don't know, 14 days or something like that. And we have a new skeleton every, I don't know, what is it, 12 or 14 years, something like that. So anyway, it's all remanufactured and rebuilt in order to stave off the second law. But once you die, there's no more machinery putting it all back together, and it just falls apart. And if bacteria get to it, game over. It falls apart really fast. So, but how fast does it fall apart? How long can it last? So these are the questions that archaeologists actually have been answering. And here's their answer. So I pulled this off of the literature. We have a for bone collagen now, not just like blood vessel collagen, which is basically skin collagen. Um, we have an activation energy, or energy of activation, as they say in biochemistry land, of 173 kilojoules per mole. So that's a way to describe how much energy it takes to break this thing apart. And now at the bottom of this slide, we have a standard form of the Arrhenius equation, which we all learned in freshman chemistry 2. And the Arrhenius equation allows us to convert the decay rate of a reaction from one temperature to another. And so this way we can perform the decay rate of collagen at a high temperature, and then so that we can do the thing within our lifetime. And then once we get those results, we convert the decay rate to what that decay rate would be at a lower temperature, a more reasonable Earth surface temperature, for example. And so those are the experiments that that archaeologists have done, and that we have replicated. And I'll show you that real quick. So here's one way to express the decay of collagen in bone, and you can see the graph on the top, and across the top says D0, D1, D2. These are days, so day 4, day 8, and by day 10 or so, there's no more collagen left at their experimental conditions, which is high temperature. So that's our best proxy for time. To accelerate the time it takes, you just increase the temperature. And and then you have a looks like a polyacrylamide gel electrophoresis result showing the collagen protein in little bands in their experimental results. So that's just one of many papers that describe the decay rate. So it does fall apart. It does last a long time. Think of the Dead Sea Scrolls. That's collagen. It's parchment, so it's skin, which is mostly collagen at that point, like 97+%. But stuck in caves for 2,000 years, so it can last thousands of years. Well, here I am with the late great Dr. Kevin Anderson, and he's the former director of the Van Andel Research Center in Phoenix, and he used to work with the Creation Research Society. So we partnered up on a research project where we're replicating the decay rates. So we're using a new technique to do that. So you can see behind us we have 3 water baths in the lab. This is a makeshift lab that they had. They've got finer digs now. You can go visit, I guess, there at the University of Phoenix. No, where is it? Oh, Arizona Christian. Sorry. Anyway, so we have 3 different temperatures. So one is running little vials full of powderized bone and shards or fragments of bone. And we did this experiment 3 times, once with porcine, which is pig, once with bovine, which is cow, and a third time with cow at very low temperatures. And we took a year to do it. And we're still gathering the results from the last one. Anyway, so you have to do it at 3 different temperatures so that you can put those on a plot. And so you can draw a line through those 3 different dots, and that's called the Arrhenius plot. And from that, you get the slope of the line gives you the energy of activation, and then we can generate the decay rate for collagen. And we did it. And in fact, we published our results way back in January of 2023, not too long ago. And so here's one of the figures in our paper published in the journal Applied Spectroscopy. So we have hundreds of spectrographs, that's the graphs, and these are just representatives of them. There's one particular peak on the graph that diminishes over time. And as a parallel technique, we also used a special imaging technique that is in the colorful columns there. And that shows wherever the red color diminishes, that's where the collagen is diminishing in these experimentally decayed bones. So we have an energy of activation also, and ours is even lower. So our results show that bones don't last as long as even this paper said, published in 2011. And they did a similar experiment. They used a different technique to measure the collagen integrity. But here's their conclusion. It's 173 kilojoules per mole. So that plugging in to the formula, 10 degrees Celsius, you're going to get between 200,000 and 700,000 years before the collagen falls apart so much that you can't even detect it. Now, this is an optimal burial environment because you don't have radiation, you don't have germs, and you don't have pH fluctuations and all the different variations that you can imagine. Most of which accelerate the decay, maybe all of which, certainly most. So that's kind of what we're looking at. So you can see this is a big deal. So on the one hand, we have the science of collagen decay. It's repeatable, it's observable, it's measurable, and it keeps telling us the same story. This stuff does not last 1 million years. It just doesn't in optimal conditions. Now, that's not a creation guy. These aren't Bible people. In fact, I met them and they're not. These guys are leaders in their field of collagen, ancient collagen, and this is what they're saying. So I can just take their word for it. And if you want to join me in that, you're welcome to. So according to them, and we've confirmed their results with our new techniques, it doesn't last 1 million years, guys. I mean, unless you have your bone like in some sort of a liquid nitrogen vat, that would help last longer, but it won't last. Of course, the cool temperature won't last on Earth's surface, so it's totally irrational to invoke that. But that's what you'd have to do to slow down the relentless effects of the second law. Here we go. Let's tour the world and look at the question: is there more than just that one T-Rex with collagen in it? And not so we have the morphology or the shape of the collagen with fibers, but we also have biochemistry indications, and we'll look at some of those. So we got the biochemical and the morphological evidence that it's original to the bones themselves, although that's really not possible in the mainstream perspective that I once clung to very tightly until a friend of mine challenged me. Here we go. Psittacosaurus. It's fun to say. Psittacosaurus. This is in the Gobi Desert, and here's a publication in 2008. And so this is kind of like one of these natural mummies, like like it resembles a mummy that you might see in an Egyptian coffin or something. But this one's locked in rock, as you can see. And in this case, they weren't even looking at bone collagen, but they're looking at skin. And they section very carefully a thin layer of that skin and put it under a special microscopical technique. That is a word, by the way, microscopical. I had to practice that one, called cross-polarized light microscopy. And so that's what gives it you've heard of, I mean, you've used polarized sunglasses. So if you take one polarized lens, orient it one way, take a second polarized lens, put it on top so that the light goes through this column, but you orient the second lens 90 degrees to the first, you do what's called extinguish the light. So the light coming through this column, if you put that in a tube, it'll be black at the detector. So anyway, so if you have light coming up from below, going through the first polarizer, then it's going to be it's no light's going to come through unless you put something in between the two polarizers that shifts the light, that twists the light just enough so that it will make it through the second polarizer, oriented as it is 90 degrees to the first. And that's exactly what certain crystals do, and that's exactly also what fresh skin does, because that's what collagen does. So collagen protein, as you saw with that ropey structure, collagen is deposited in like a crystalline sheets and layers, and that's what accounts for the fact that you can see it in this image, and that's what accounts for the fact that it comes through in these beautiful shades of gold and blue and purple and even orange and a little bit of red. So anyway, there's that evidence. So we have, it's like actual dinosaur skin, not just skin impressions, which are rare enough, but this is actual skin stuck down onto the bone like a mummy. And there's evidence here that it's actual skin. It's just old, decayed, rotted skin, but it's not fully rotted yet. And so we wonder why. Now, where else? What about this dinosaur, Lufengosaurus? There's a silhouette of it showing the comparative size, published in Nature. And these guys used they used cross-polarized light, but they used a fancy version of it. I'll just put it in that. So we have multiple publications, and so these are some of the top journals in the world. And what did they do? They looked inside the egg of this dinosaur egg, and most dinosaur eggs that I've seen, including mine, it's just infilled with clay. The original dinosaurs rotted or gone, but in this one they found the embryo still in there, and it looked pretty fresh. So they cross-sectioned the femur bone, and you could see cross-polarized light is doing something, and it's twisting the light. So they concluded that that we have complex proteins preserved in our specimen, and by far the most abundant protein in bone is collagen. And so it makes, and we already know that collagen bends light. It's called birefringence, and it has that property in fresh bone. So and they did other techniques, tests to verify that one. So we have it in the Orient. We sure do. And it's not just dinosaurs. Here's a fossil turtle. And this fossil turtle, you can see the mud sort of caked beside it. And some of the mud, it's a yellow mud, but the bone is even yellower if you look really close. And this was published in the journal Bone back in 2012. And the study authors investigated this bone. And one of the things they were able to do is dissolve the bone. And when you put the bone in weak acid, the organic uh, um, some of the organics that might still be in there get liberated because the acid um, dissolves the mineral part of the bone, which is bioapatite. So anyway, some of you did that experiment. It's really fun to do. You just put a chicken bone in a jar and fill it with vinegar and wait a month. Then you pour out the vinegar after a month. And you pull out your chicken bone, and the only part of the chicken bone left, there's a bunch of sediment at the bottom, and that's all the mineral that's dissolved. The only part of the chicken left is the collagen protein. So you end up with a perfectly rubber chicken bone. I need to do that and just have one hanging around, although it's a little bit smelly. My wife might not appreciate it, but anyway, they did that something similar. And look at all these are bone cells. The cells are still there, and the cell membranes are still intact, holding these cells together. And they even have these thin extensions that in life they penetrate the bone, and they even communicate with neighboring bone cells, osteocytes, what we call them. Anyway, they found osteocytes still in there, and also in dinosaur bone, as you'll see. Not just dinos, but turtles. Now, here's a replica of a famous fossil named Caudipteryx. Some of my colleagues call it a feathered dinosaur, but there's other experts who call it what they call a secondarily flightless bird. So it may be a bird that just the Lord designed it to produce a generation with smaller wings, so it couldn't fly, kind of like the flightless cormorant, which we think descended from the cormorants that can fly, the varieties that can fly. Anyway, so maybe that's what Caudipteryx was. Regardless of what it was, it's also from the Orient, the famous G-hole biota, which it's got salamanders and all kinds of insects. Um, so we have. Think about this. I mean, it's birds and salamanders and insects. Like insects can walk on land. Salamanders are in the water. Birds are in the sky, and they're all washed into the same pit. In the pit, or I say pit, I mean low area, and it's a broad and extensive low area, and it's exquisite preservation is what we call it. Sometimes that phrase means um, that the original organics, like the organs, soft tissue organs, have been preserved as minerals, so that minerals have replaced the shape of the organ. So we have to be very careful because the literature will say remarkable preservation, and they'll say like we have a fish brain, but it turns out that it's all phosphatized. It's just a phosphate type mineral that's taking the place of that a liver or whatever it was. And so, but we're not talking about that here today. Those are interesting because it gives us the shapes of organs of some extinct creatures. But what we're talking about is the um, original biochemistry, original tissues, and that's what they found in Caudipteryx. This is a little bit strange to look at, but it's the best image I could pull from this particular publication. But Bailul, she's the lead author, and um, she did a stain. She did two different stains for DNA, and they stained positive. And it was she found chromosomes in special cells and chondrocytes. And so these chondrocytes are like cells that manufacture cartilage in joints. So if I can go back to this, um, hopefully you can see my mouse. But they looked at the joints where the two bones meet and articulate, and that's where we see these cartilage um, cushions, and inside that is where she found chromosomes, or at least stains in chromosome that were the shape of chromosomes in those cells that happened to have been doing mitosis when the creature died. Um, anyway, so you can look up that paper and study that yourself. So people ask me, what about DNA? Is there dino DNA? And I'm thinking we have some. I'm thinking we have some, and here's one of several papers that have been published just recently with DNA. I lean toward calling this one bird, but that's up to you to decide what you want to classify that guy as. But that's the Orient, guys, and so we have apparently original organic tissues coming out in major peer-reviewed journals um, all over Asia. And now let's go to Europe. And here's one published in 2011. This one's a free online one, so that we can use these images and not have to pay royalties and get in trouble or anything. So this is safe to use. Anyway, so the authors did several different analyses. You can see on this mosasaur. So that's an extinct marine reptile. Just think of a 40-foot long, I guess, a snake type creature. And you have osteocytes there across the top, and then you have in box G an osteoid, which is which includes ECM, or extracellular matrix. So you know those osteocytes, that's the cell with all the different projections, the skinny extensions. And what are those doing? They're manufacturing bone. They build bone, and they put the bone in place. And so they're building bone constantly while we're alive. And what is bone made of? It's a mixture of collagen and um, other small amounts of many other proteins, but mostly collagen, and then mostly biomineral, hydroxy apatite. So then in box H, which I think is so, I would show this to my wife and kids, and I'd just be jumping up and down. Look at box H, look at that, and they're like, "What is it? Some sort of a ghost?" I mean, it means nothing to me. So I'm taking my time explaining these things, but that's actual floppy, that's protein floating in a solution on a glass slide underneath a microscope. It's just stunning to think that that's material that's still there, but that's what they published. And then, of course, bottom right, you can see the cross-hatched pattern of deposition, reflecting the bone structure there inside this mosasaur. So anyway, they concluded this primary organic molecules, including collagen. Now, primary means it didn't come from outside; it was in the bone itself, like the thick, dense part of the bone. So it's primary, not secondary. Including collagen or its degradation products, are preserved. They are preserved in these tissues, and there's the accession number for this particular fossil there. What about, let's go to Poland. Okay, so in Poland we have a T-Rex-like dinosaur, Tyrannosaurid, called Tarbosaurus, and wait, this is published back in 1998. What were they looking at back then? Electron micrographs. And if you look at the box top left, you'll see this blood vessel. That's how they interpreted it anyway. This blood vessel that's coming out of the bone, and they've partly demineralized the bone, and so the mineral part dissolves away, but the biological part is still there. And now it's been there for thousands of years, so it's puny, and that's what we call it in Texas. Man, I feel puny. So it has lost a lot of its integrity, and then but it's still there. And then they interpret box 9 as a blood clot. Anyway, it says erythrocytes, so that's red blood cells, and you have these biconcave discs. Actually, in dinos, it wasn't biconcave; it was just like a disc, and they had nuclei, so nucleus in there that they've looked at under the microscope. Mammals, we have biconcave discs, and there's no nucleus in our erythrocytes, but reptiles were different. So anyway, um, erythrocytes and their content of iron must have been present in hemoglobin. So they're speculating that it's hemoglobin. They don't have a direct test for that, but other specimens have been directly tested, including the very first image I showed you of the 2005 T-Rex, published in the journal Science. Direct presence. Okay, so we're still in Europe, right? And we're looking at fresh fossils in Europe, our second continent. And here's a beautiful piece of art there in Spain, Barcelona, on display. It's not on display really, but it's there. It's General de Palitat. I don't know how to pronounce it in different language, but I think it's a private building or a government building. Anyway, this depicts St. George slaying the dragon, and the artist chose this particular creature as a dragon. Is it fictional, or is it just happened to look exactly like a Nothosaurus? I mean, the same body proportions as we know from fossils, the same characteristic mouth shape, and the same characteristic long teeth that extended past the jaw there. So Nothosaurus, that's the spitting image of it. Make of that what you will. But here's actual Nothosaurus blood vessels published in 2016. This is free online. Also, you can look up the reference at the bottom. And so you have a network of capillaries that in box B, and they've liberated these from from bone by just dissolving the mineral part away. So they applied, I don't know, a dozen different techniques. They're using microscopy, and they're using all kinds of spectroscopic techniques, FTIR, and I mean Raman spectroscopy, and you name it. They put it under all their different techniques, and they concluded that this is original blood vessels, but they found a mineral called goethite, which is rich in iron, and it's pretty common in fossils. It can even form these iron balls that we find when we do digs in Montana, goethite balls. And anyways, so it's pretty common, but you could see goethite attached to some of these blood vessels there in box A and some of it in B. And they suggested maybe these blood vessels got coated with a layer of goethite on the outside, and that's what's preserved them for all this time. Now this is a nothosaur, so it's like it's in rock layers below the dinosaurs typically, and so I'm forgetting the details on this, but I'm thinking Triassic layers. So there's a few dinosaurs in Triassic layers, but anyway, the conventional age assignment for Triassic is over 100 million years, and yet blood vessels down there too. So this is pretty stunning, and it's in Europe. So they proposed that minerals are doing it. Minerals are doing it, preserving these. So that's really one of my areas of interest, is how can we get these proteins to last even longer? And so they're saying maybe that iron from the mineral sticks to the blood vessel. Of course, that does have a lot of problems. For example, where are you going to get the iron, and if and how are you going to get the iron there? So that if you, let's say you do have iron, rich mineral like goethite, to transport that in, it has to be transported inside the bone, and to do that, you have to bring in water as your vehicle of transport. And the water that brings it in is going to actually interact with the proteins themselves. And that's what's going to facilitate even more chemistry. So it adds more problems than it solves. Certainly, iron would help to keep away microbes. Microbes notoriously or famously don't like a lot of iron. So anyway, I think that's what's happened here, but it still doesn't seem to solve, like, at all, the orders of magnitude difference between what we'd expect to see, um, according to the lab bench decay rate studies that show that this stuff can't last a million years. Uh, so that's what we want to do next, is we want to add goethite into our test tubes, into our bone powder, and see if we can see if iron actually, see what effect iron has. All these hypotheses on how to get this stuff to last for millions of years, none of them have been empirically tested. They're all, this might work and that might work. And I mean, people are running with these hypotheses. None of them have been tested. So we want to be the first to actually test them. So anyway, but we need your money for that. So uh, maybe you'll join the club and find inspiration in helping us do that. So what about this one? Here's a turtle from Europe, and what do they say about it? Biochemistry. uh, Biochemistry is still there, and coloration. So we have lots of fossils um, with pigments, melanin and such, still in there. In fact, lots of them have melanin. Don't have a decay rate for melanin, so I can't make a strong argument for or against it. But what they found in this turtle is, so wherever it glows in this particular immunohistochemical technique, wherever it glows, you've got an antigen binding an antibody. So they raised antibodies to feather, just chicken feather, which is mostly keratin protein. And there's lots of fossils that have been found with keratin protein, but I can't even include them in this talk because we don't have days to talk about this. And they found it in the fossil. They raised hemoglobin, and they found it in the fossil. They raised hemoglobin from alligator, and that matched better with the turtle than did the hemoglobin antibody raised against Struthio, which is I think ostrich. And then tropomycin, and they found it. So it's glowing in all in that second column, glow, 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 glow. And so this shows that we have traces of the original of these biomolecules still in that turtle there in Europe. Here's a stunning one. This one is just look at these fibers. Anyway, sorry, I don't have the date on that reference. I got to fix that. Sabelides is a sea worm, and by the way, it's a sea worm that makes its own like casing that it lives in. It anchors this casing to the bottom of the sea. It's one of these worms that lives into the casing, and it has this like feather-like projection that comes up, and if it sees a shadow or it detects a shadow, it'll pop back down into its casing. Well, what's the casing made of? The casing's made of a mix of proteins and chitin, a type of material that's also, just think shrimp shell, chitin. Okay, so anyway, they said the body wall of Sabelides cambriensis, because it's a Cambrian fossil named after the Cambrian rock layer. This is the lowest one we have fossils in, by and large. It's got an evolutionary age assignment of over 500 million years. That's half a billion, and yet it's got original chitin structural protein composite is still there. And they took an electron micrograph of it, and they found that the elemental composition of those fibers matches, and the 3D shape of it matches also the morphology of today's tube worms in the sea. Anyway, that's just fantastic. So we're finding it in lower layers, we're finding it in middle layers, we're finding it in upper layers, as though all these layers were freshly deposited and young enough to have original proteins in them. And that, by the way, is what the flood model tells us. That's what the Bible even says. It says that the whole heavens under the whole earth was covered with water. So we think that the whole stacks of rock, thousands of feet of rocks in a lot of places, all of it deposited in 1 year. And that's the flood model, which means that you might expect to see original biomaterial in lower layers, middle layers, and upper layers. And that's exactly what we're finding. You also might expect to see this worldwide. Okay, we've got it in Europe, we've got it in Asia. What about South America? Well, I wouldn't go there if there wasn't something. What about a titanosaurid eggshell where they detected the presence of ovalbumin, which is a specific vertebrate and in fact, an egg specific protein that vertebrates make there? And what about a bird, fossil bird from Argentina? And here we go with the glowing business again with the antibodies. And so they're finding bird collagen in that fossil from Argentina. And there's more, but let's get to North America, my home. Here we have the Green River Formation with outcrops in Wyoming. And here is half a lizard. It's the back half. So something ripped this lizard in half and slung it in mud. And then this mud was deposited in layers, famous for its fish. Anyway, so these researchers were looking at the lizard fossil that they have, um, embedded in shale. And they were just marveling at the detail of these scales. So they zoomed in. You could see the little zoom in box, and each scale is dark. It's like a dark circle there. And so we're asking ourselves, what makes it dark? Like, why is it dark, and not what material is it that makes it dark? And they found keratin. So then they were like, and by the way, so the structure of a reptile scale, even today, it's just imagine like I don't have any artifacts to just imagine a disc made of keratin. Like imagine a tiny fingernail, something like that, and you wrap that fingernail in skin. Okay, and then you bind that skin to the next scale and the next scale. So it's like a little skin covered armor plate, semi flexible armor plate. Anyway, they found the keratin still in there, and they proposed that minerals had come in, attached themselves to the keratin, holding it all in place. and locking down those keratin scales for 40, I think 40 to 50 million years would be my estimate. Not remembering this particular paper because I've read over 100 of them, which I'll show you here in a minute. Anyway, again, you have the problem of the water that would bring it in would be the same water that would bring in the oxygen and the and the biochemistry that would tear apart. Anyway, um, it has its problems, and it has not been adopted by the larger community, mineral lockdown that is, as a means of preservation. It's pretty far-fetched, guys, in my opinion.
Mary put some fragments of the bone in acid to dissolve away the outermost layer of mineral. But the acid worked too fast, and all the mineral dissolved away. Being a fossil, there should have been nothing left, but there was, and it was elastic, like living tissue.
This is the piece.
No. She showed us video she took under the microscope.
That's really what happened?
Yes.
That's the dinosaur bone?
Without mineral now. That's what was left.
It looked like the soft tissue she would have expected to find if it had been modern bone. This was impossible. This bone was 68 million years old.
So you see this and you think, what? You say,
I didn't want to tell anybody.
You'd be ridiculed,
Yes.
right?
And so I said to my technician, okay, do it again. I don't believe it.
And yet, in sample after sample, they were there. Things that looked suspiciously like flexible, transparent blood vessels. She finally mustered the courage to tell Jack.
She said she dissolved the bone away and there were blood vessels. And, I was like shocked. I mean,
How could that be?
How could that be? That's right.
The things Mary was finding inside dinosaur bones,
Look at that.
blood vessels, and even what seemed to be intact cells, pose a radical challenge to the existing rules of science, that organic material can't possibly survive even 1 million years, let alone 68 million. Mary, Jack, and their team published.
Well, of course, I was interested in this, and I've been able to find resolution that I think my secular colleagues, like the one they're interviewed, still struggles to find, and that resolution has to do with you don't have to challenge it's not a rule of science, existing rule of science that says that these materials can't last 1 million years, let alone 68 million. It's an existing experimentally derived result of science that says that. So they're trying to get around it by calling it a rule, but anyway, we do have a solution, and that is to, instead of challenging the science, we could challenge this concept of deep time. But that's a place that too few of us are willing to go. I'll just leave it at that. But if these fossils were buried in the flood, then it would be only thousands of years ago, and then that would explain all these results that we see, including from hadrosaur. I took this image of most of it, what I could fit on the camera, at the Denver Museum of Natural History, and it represents, I guess, some kind of a hadrosaur. This is actually Brachylophosaurus hadrosaur, published here in 2009. What a great paper this was. Collagen protein sequenced there, and blood vessels again, and bone cells again. Where else in the U.S.? How about Triceratops blood vessels, ECM, extracellular matrix? This stuff looks fresh and flexible. It even made its way into the movies, where the kids Zach and Gray are talking in the 2015, Jurassic World, and I've got this little chemistry diagram up because part of the movie dialogue, the little brother says, iron can preserve it, and that's why we have the DNA. So this is all speculation, but they grabbed this from a paper that had just been published and put it into the dialogue of the movie. The paper said that iron can help these preserve by cross-linking them. So you have a protein here, protein there, they cross-link and they make something that's no longer a protein, but it's bigger. It turns out that in reality, the iron may do that, but many more times it will actually do destructive chemistry instead of constructive chemistry. It's just random chemistry, so it's going to destroy more than it builds. And it's also going to promote oxidation. That's what brings these two together, an oxidation reaction. But that should have, if that was the case over these eons, oxidized, for example, one of the amino acids in protein, methionine, which has that sulfur that loves to do chemistry, and oxygen would stick right to it and change it from methionine to methionine sulfoxide. But what did they publish in their protein sequence? They published, hey, we found methionine. So on the one hand, they're saying iron preserved it, but we know iron would have brought in the oxygen, facilitated the oxidation reaction. And on the other hand, they're saying, well, we have unoxidized, still fresh looking methionine. So there's still mysteries that they need to solve and that we need to solve. Let's do a few more. What about, let's go to Kansas, because everyone knows this is a mosasaur in Kansas, and everyone knows that there's a big ocean right in the middle of Kansas, right? I think this is a flood fossil. The Bible says that the waters covered all the high hills under the whole heavens, Genesis chapter 7. I saw this described in the literature, and I was like, here we go. I got to read through this paper. I'm just kind of interested. And the more I read, the more I liked it. And they finally got to this part where they're investigating a little bit of red in its chest cavity, the presence of hemoglobin. There it is. They think it represents the residues of visceral organs. In particular, they said that the positioning of this red patch matches the location of where a heart is positioned in a streamlined creatures like dolphins. So they think maybe this is the residue of its heart. Lo and behold, I was in the LA County Museum of Natural History. This very fossil was on display. I wasn't expecting it, but I was jumping up and down when I saw it, and everyone else in the room like backed away. I don't know why, but there I am pointing to the still red colored heart residue. That's what they said about it. And you can now see that there's skin scales are there too, and it's like mummified here again in fossil excavated in 1967, but transported to LA for display long ago. And then now that new techniques come online, we can use these non-destructive techniques to do all kinds of analyses and look for original organics, and they found it there. Well, now we're going to transition to some of the work that we're doing and look at this. We're interested in the Permian. We're interested in Mississippian, Pennsylvanian, Permian, very low rock layers. These have age assignments on the orders of several 100 million years. And here's this creature called Ariops, at least an artist's rendition of it. Ariops. Just imagine a 7-foot salamander, something like that. And here is my colleague Jim Soliday's lab. You can tell he's a very highly organized guy, but he does great work. Award-winning microscopist, and he has trays and trays full of thin sections of dinosaur and other fossil bone. And then he uses that microscope there. You could see it's a cross-polarized light microscope. So, are you ready to see some pretty beautiful images? Here's an image of our Ariops, and this is a thin section of bone. So light's coming through the bone, and you can see these big black holes. Okay, so those are like empty spaces. It's black because the light's extinguished. I'm sorry, that's not it. It's black because it's occluded. There's something in the way. But then you see you see like bone structure in here. These are osteons with the wrap around. The collagen is laid in these rings, and if you do a cross section through the rings, you can see that. And there's a big mineral that has infilled this where this was probably a blood vessel in here. Now we have mineral that has come in and filled part of that space there in that ancient fossil. Now we have it under cross polars. So you could see where it's extinct and where it's not. And what that does is it reveals the regions of that bone that have collagen in them, and it's twisting the light so that it's bright, it's yellow, it's purple. It's also cracked. You could see those cracks in there, which indicate that it's ancient bone. And something is twisting the light, and we're pretty convinced it's collagen. And then Jim adds a red filter, and we get this continued effect that further illustrates and highlights what we're interpreting as the presence of collagen in certain regions. And this one, it's wrapped around the osteons for some reason, and the collagen is all decayed away in all those dark purple areas, but where it's gold, you're looking at collagen. To confirm that, you just rotate the microscope stage, and it's going to change the color. So I'm going to rotate this stage and then show you the color pattern changes. So this is 90-degree rotation, and you could see where it was gold, it turns into purple and goes through the rainbow, and that's an indication of collagenous remnants embedded in the very matrix of the bone. They're very ancient bone. Here's one here's a video I took of it, so you can see this process. There's a big white calcite crystal inside the bone. This is Edmontosaurus, a thin section. Now you could see it changing color from gold through blue to purple. And that was an exciting day to see the old cracked bone still has something doing by refringence. Okay, so we interpret that as collagen. It's a pretty safe interpretation. And here's another hadrosaur bone that I worked on for my thesis, and the little red dot with the circle shows that a new technique, newly applied to fossils anyway. I worked on that. Second harmonic generation imaging. It uses a laser to have it has a peculiar effect with just collagen. So it's a very collagen specific result there, and we found a little bit of collagen left in that one. There should be none if it's as old as the mainstream assertions suggest. Okay, and now we're working on a sacrum, and so this is an Edmontosaurus sacrum that we just got accessioned at the University of Liverpool, where I got my PhD. I still have an association there, an affiliation there, so I'm still doing work with my colleagues at the university. We have a modern horse bone for comparison because it's the big dinosaur bone, and we've been digging into this dinosaur bone, and here's an exciting new result. We're trying to get this published, but you could see on the top you've got these green peaks. This is indication of collagen, which when it fragments and goes through the mass spectrometer, you get these specific masses that are detected, and this is like a signature of collagen. And on the bottom with the red, we did the same thing with a turkey bone, a modern turkey bone, and we got the same masses in the same positions, but in a much greater amount in the turkey bone, which makes sense because it's modern turkey. But these little green peaks are just a real firm confirmation of collagen that's still in our dinosaur bone. So one day we'll be able to hopefully add that to this list. We have over 120 some odd now that are, and you can see on the bottom right, the URL. This is free. Anyone can look at our list that we've collected of original biomaterials in fossils, and these secular peer-reviewed journals. Guys, we have it in Asia. We have fresh-looking fossils in Europe, South America, North America. I think the only reason we don't have much in Africa is because we don't have a lot of scientists digging around in there. I think Australia has some. We just haven't been published yet. So it looks worldwide. So you need a worldwide cause to produce a worldwide effect. The flood does that for us. You need a recent cause to produce a fresh-looking effect. The flood also does that for us. And so these collagen and even chromosomes are fresh fossil features, and they are worldwide. And they confirm for us what I finally came around to believing wholeheartedly, which is the Bible itself. And this is what 2 Peter said about scoffers who would come in the last days, and certainly my professors who drilled into me, you got to believe in the age of reptiles, and you got to put away your Bibles because those Bibles are a bunch of junk. That was the upshot of what they were saying. But Peter says when they say these things, they are willfully ignorant that by the word of God the heavens were, and the earth. So he spoke it by a miracle, and that's how we have the place that looks so miraculously constructed with all of its specificities and all the creatures and everything we need to live and move and have our being. And they're willfully ignorant that the world, that the water out of which that first world was made, that that whole world that existed perished, being flooded with water. In other words, the two miracles that modernists deny, creation and the flood, the two miracles that I denied because that's just the air we breathe, those two miracles, um, the creation and the flood, are the most attacked historical events. But I'm studying the real world and I'm finding evidence that fits creation according to kinds, dinosaur kind this, dinosaur kind that, bird kind this, kind, and recent looking with all these fresh fossils as though it was flooded recently. So the flood really explains the data, and the creation explains the design data also. Well, you can access more of this kind of exciting results. We try to put them up on our website at the Institute for Creation Research. This is an older version of our web page. We've given it a paint job since then. I need to update this, but that's all for free. So that we have dozens of articles on this, totally free. Of course, books also and videos. Speaking of which, we have a family book, color pages on every page. And if you like the decay books, this is one of those. And we have Guide to Dinosaurs, Guide to Creation um, Basics, and there's a whole series of guide to. And we're making another one. We're producing one. Um, I don't know when it's going to come out, but we want to make one called Guide to Human Origins. That'll be fun. And then we have a 4 DVD set, TV ready, TV quality. We paid a lot of money to get this produced professionally. Go on location to Dino Digs, and we look at different the soft tissue issue, as well as dragons, like where did those come from? We explore those issues. And then this little booklet I wrote, Dinosaurs in the Bible, explains some of those that's actually in the Bible, like what really swallowed Jonah, and what was the serpent that was lifted on a pole to which the Israelites looked and were saved, and things like that. And then Creation Basics and Beyond, which explores all kinds of issues. Now, I'm showing you these physical books, but you can get all these digitally. So you just go to store.icr.org and just buy anything you want there, and just get the digital download. And it's like half the cost of the physical print, and you can put it on all your devices, things like that. So Creation Basics and Beyond is what I recommend for every college student going into or coming out of a college, especially because you can just go right to the chapter of interest. So if you want to know about the Ice Age, go to that chapter. If you want to know about carbon dating, go to that chapter, and get some answers that are scientifically responsible and biblically sound. And then we have our free magazine, Acts and Facts, which you could also sign up for. I'd encourage you to do that at icr.org. It has been a privilege, and I appreciate all your attention and putting up with me tonight. Hopefully we had some fun and looked at these fresh fossils with fresh eyes.
Good. Thank you, Brian. That was really exciting. I'm very impressed with the information that you've presented. The information that you've compiled in the Google Docs area, we're going to put that information out in the comments or in our material so that people can look at that and be able to reach out and grab it too. I've seen it before. It's really powerful stuff. All the information is hot linked so that you can quickly get to the papers that are out there. And one of the questions I have, Brian, if you can hang around for a little bit longer, we've got several questions. But you indicated that the trends that you saw in the secular information really did not show a pattern with being shallow in the geologic or fossil record or deeper in the record. And that's exactly what you would expect. So I'm excited about that. Did you see anything else in your collection of your review that would be interesting to share with the audience?
I would say yes. In fact, we published a paper in the journal Expert Reviews of Proteomics in 2019, the results of this literature survey. And so you can find that paper, and It's called Proteomes of the Past. So that's the paper title. But I found 3 trends, and one was that the original tissue fossils don't respect the layers, as you have mentioned. In other words, you find them. You could find it in any layer. Even the bottommost layers. And then the next trend is, I mean, is it just dinosaurs? No, it could be found in any taxon. So it doesn't respect taxa. So you could find it in a turtle or a monkey or a frog or you name it. You could find original biomaterial still in there. So in other words, we're kind of like trying to find patterns, but it's like there's no pattern. It doesn't respect taxa. It doesn't respect geology, nor does it respect geography, as I've tried to show in this slide presentation. We might find it on all the continents. We just need to get the Australians to start digging around some more and looking.
Awesome, that's exciting. We do have a couple of questions I'll throw out at you, and I'm going to sort of summarize a couple of these.
Thank you.
One was a discussion about what is Mary Schweitzer, who was one of the first discoverers of this. What does she believe is the presence? What's the preservation for these soft tissues? But can you? I think you've addressed that, but can you sort of generally talk about the mechanisms that they believe are popular right now?
Yeah, sure. So she published a paper that showed results of basically bathing exposed blood vessels in an iron solution, like a solution of concentrated iron, and it helped preserve those blood vessels at room temperature for several years. And the control was, no iron, and those rotted with fungus within several weeks. And so based on those results, the paper came to the conclusion that therefore, if blood vessels were bathed in iron after the creatures were buried underground, then that could preserve them for tens of millions of years. Now we see that's not a logical, that doesn't follow from the data. So nor is it rational to propose that there was some sort of a Cretaceous laboratory where where we have iron being concentrated somehow. So there's problems with the mechanism. We got to get the iron from somewhere. We don't know where. We got to get it concentrated, and we have to bathe these blood vessels in the iron. And those blood vessels are embedded in, they penetrate through bone. But even if we did all that, what we'd have iron-mediated cross-linking. It would be like it's like it mimics like formaldehyde. So we drop formaldehyde in to a living specimen or a dead organic creature in a jar to keep it preserved, so students can look at the dead frog or whatever. And so it does that. It does cross-link those things. Fine. But they're not finding the formaldehydeized or the cross-linked. They're not finding as much of that. When they actually do the protein sequencing, they're finding the actual original proteins. They're not yet oxidized. So on the one hand, they're claiming that it's oxidization that has preserved these by cross-linking them, but on the other hand, they're showing results that show that it has not been oxidized. And so it's anyway. So there's the iron preservation hypothesis, and then now there's one called the toast hypothesis. I mean, I call it the toast hypothesis because it's easy for me to remember. But these AGEs, or accelerated, I forget what it stands for, glycosyl end products. Anyway, so it's literally what makes the crust on your toast is when the bread is baked at that high temperature. It does some chemistry, and it does something similar to cross-linking. Anyway, so now they're trying to combine those two, and they're saying that I just read a preprint paper just a couple weeks ago, and that they're trying to argue that once you throw in the iron and the toast, now we're really helping to explain things. But I have my doubts that it'll pan out in the end because um, they still have the original like protein sequence that's there, and so it's not toast. It's not.
Right. Mhm.
It's not been toasted, that part of it. So they're
Mhm.
they're trying to say that the toasted part is right next to the original part, protecting it. Well, that just violates the second law by
Mhm.
pretending that just because you have a layer of soft tissue butted up against a sheet of plastic or something, that it's going to last for tens of millions of years. It would be like having a candle, looking at the candle burning down and saying, that candle has been burning for, the last 300 million years. And it's like, how high would it have been?
Mhm.
It would have penetrated, past the moon, where there's no oxygen. It's like, it's just, it's really tall task to try to get ways to get these short-lived biomaterials to last as long as mainstream, um, the mainstream requirements for it. Um, Regardless of all that, these are all the iron hypothesis, the toast hypothesis. They have not been empirically tested. They haven't. So we're waiting on that. So I'm not gonna. I'll buy it if I see the evidence for it, and I'm willing to find it. I'm willing to test it. So that's what we want to do next with
Sort
our
of a
programs.
related question, Brian, is why do you think that the secular scientists haven't jumped into a lot of these testing protocols for these hypotheses?
Oh, I can't. I would only speculate on that.
Mhm.
But if they did the tests, and if they added iron, if they did the toast somehow, toasted their bone, and then see what toasted bone does.
Mhm.
If they ran those tests, then they might not be happy with the results. Perhaps that's the reason.
Mhm.
You also have to get the funding to do and it's a very labor intensive and time intensive experiment. I mean, it takes half a year to run the experiment. That's after you get it all set up,
Mhm.
and then you have to. So anyway, it's not an exciting venture for them. I would think so. It's easier to get funding for something where you just discover blood vessels, yay,
Right.
um, which would be great too. So
Yeah. So
those are
here's
some
another good
speculations.
one. Here's another good one, Brian. Are there any carbon-14 found in the dinosaur bones? If you have original materials, uh, has anyone tested for carbon-14?
Well, we have, and we have several dozen um, dinosaur and other fossil bones that we've sent off to, I think, 8 different radiocarbon labs all told over the years.
Mhm.
We've got fossil wood, we've got fossil coal, we've got fossil um, dinosaur, fossil fish, and we just keep sending it out to the labs, and they keep sending us back the results that there's radiocarbon in there, and in measurable amounts. In other words, amounts of radiocarbon that are above the background levels that they've measured in the lab. So
Mhm.
that's significant. That means you've got radiocarbon in there.
Mhm.
Um, so um, so we're trying to get some of that, more of that published. We're meeting, as you can imagine, significant challenges and difficulties. Nobody wants to publish our radiocarbon and dinosaur bone results.
Right.
uh,
Okay. Mhm.
so
All right. Excellent. Well, we can certainly pray about that and
yeah,
continue to support you, Brian. That was a fantastic presentation. So, just in terms of the time, there are a couple of other questions, but it's about 15 after the hour. So let's wrap up. And what I'll do is I've got a little bit of discussion here that I'll start. And again, I want to thank you, Dr. Thomas, for all the information you've shared during this fascinating discussion. As a reminder, we want to encourage anyone watching to check out the Logos Research Associates website. On the website, you can learn about the various research efforts of all of our associates and ambassadors and find information about their publications. I particularly want to highlight our updated donation page. When you visit the Logos website, you will notice that our donation page includes several panels that each describe a current or planned research project sponsored by the organization. This donation portal gives you the unprecedented ability to directly support the creationist research that interests and excites you. For example, the Paleozoic Deep Cell Project aims to discover and document even more examples of soft tissue structures throughout the geologic column, furthering the work done by Dr. Thomas and other creationists in this field. In the near future, we also plan to use this portal to provide donors with project-specific updates so that you can stay connected with the creation scientists whose work you have enabled through your generous giving. So please take a few moments, browse this page, and consider supporting one or more of these exciting projects. While you're on the website, please be sure you have subscribed to the Logos email list so that you can receive notifications about our other upcoming presentations. And when you subscribe, you will also receive the Creation Evolution Headlines newsletter. We want to let you know that a recording of this video will be made available on our website, as well as YouTube, our channel for Logos Research on YouTube, as well as Facebook Live. So you can share it with others who may not be able to join us live tonight. Our next presentation will be on Wednesday, May 17th at 9 p.m. Eastern Time. Our presenter will be Dr. Jeff Tompkins, also with the Institute for Creation Research. He will discuss his research and show that the human genome and the ape genome are not 99% equal. This presentation is titled Monkey Business and the Chimp Genome. I'm now going to close this in a short prayer, if you would join me. Dear Heavenly Father, thank you so much for Dr. Thomas and the work that he has done and the impact he has had on so many people. Thank you for his dedication to this new and exciting field of research. Thank you for these broadcasts and their ability to reach so many through the internet. We also pray for Dr. Tompkins and his upcoming presentation. We pray all these things in the name of Jesus, amen. Well, thank you all for joining us, and we'll see you in the next presentation.